Hello everyone, my name is David Foltz. I'm a senior malacologist and astacologist at Edge Engineering and Science, and today I'm going to be talking to you about unraveling the distribution and genetics of Cambarus species C in Virginia, preliminary results and focuses moving forward. So let's meet the complex. Here we have the acuminate crayfish, Cambarus acuminatus. And a little bit of background. So Cambarus acuminatus was described from the Saluda River in South Carolina by Faxon in 1884, and then over time, as collections continue to expand up the East Coast, variability within the complex the animal was noted, which eventually led Hobbs to suggest it was a species complex and then dubbed it Cambarus species C. Since then, multiple species have been described from the complex. However, issues via taxonomy, conservation, and distribution persist. So how do we come into this? Enter Cambarus species C from Pennsylvania. It has a fairly disjunct distribution from the rest of the complex, and Fish and Boat Commission wanted to know, is this population introduced or is it native? And basically by asking this question, a giant can of worms was opened, because due to how the complex has been treated so far, you cannot answer the PA question without first tackling the complex itself and filling in knowledge gaps. So Fish and Boat provided the original funding, and then through various other agencies, supplemental funding was obtained, which eventually led to this project working its way all the way up the coast. So preliminarily, Williams et al. In 2019 published a redescription and circumscription of Cambarus acuminatus from the Saluda River in South Carolina. What this did is it treated the animal down there and determined that acuminatus from the Saluda was acuminatus, and it only occurs in the Saluda. So everything else all the way up the coast is just in the complex and currently needs description. That same year, Perkins et al. Described Cambarus franklini from the Watauga. So let's look at these animals a little bit. Aside from acuminatus, one of the first things described from it was spicatus. Neat little crayfish, lance-like rostrum, very spiny cheeks. Then you have Cambarus hobsorum, described by Cooper in 2001. Big beefy animal, very unique looking rostrum, narrow areola, very sandpaper texture heavy sides. Then you have hystricosis, which is this individual right here. It has multiple cervical spines, and Cooper stated that it was due to this, it was probably closely related to spicatus due to the spines on the cheeks. Then you have John 2006. Big animal, kind of plain looking, very elongated rostrum, small, straighter claws. Some of the more recent animals, Cooper and Price, 2010, Alder minorum, this highly contrasted yellow and black individual. Very parallel margins on the rostrum. Then over here we have franklini, this green, yellow, red individual, rainbow colored tail from the Watauga. Then down here, acuminatus, which is just kind of plain Jane looking brown and green. So that's the basic background, but before we jump into this, we have to pull the veil back even further, because what I've given you so far is overly simplistic. In order to answer these questions posed and properly treat the issue, we had to do this massive undertaking of sampling from South Carolina all the way up to Pennsylvania. And as you can imagine, that took a lot of effort. So our methods were to obtain at least one sampling site from each Huck 10 throughout the entire range of the Cambarus species C complex until we could no longer find the specimen in the next Huck 10 downstream. And then no X number of seine hauls or dip netting was performed, just whatever got us the animals. As you can imagine, sampling in mountain streams in Virginia is very different from sampling in low gradient Piedmont streams, say in South Carolina. So what we found best was to use a combination of all seine hauls, dip nets, snorkeling, hand collecting, whatever got us the number of individuals per site that we would like. So kind of how it went is we would start a day, I'd have my coordinates here, we'd just drive around, we'd get the animals, and then they would be vouchered into genetics grade ethanol, and then we would check the site off the list. Here's an example of my sites. This started out as very daunting, but it was kind of gratifying to change dots from blue to green and check stuff off, but that's just for the field sampling. So on the genetics methods, we did a tiered approach where we focused on patterns across a multi-locus molecular genetic data set. Due to the density of the data set and what we were trying to do, we did 2 individuals were sequenced per site for the CO1 gene. This helped out because it kind of functioned as a quality control as well, since we're doing 2 per site to let us know if we got anything wonky. And then from that, additional subsampling of 10 to 20 individuals per clade for a nuclear gene gap DH at 825 base pairs. I'm not going to sit here and pretend to be a geneticist. This is more of Bronwyn's bread and butter. So if you have any questions pertaining to the genetics, I invite you to kindly direct them to Dr. Williams. I'm just here to give the preliminary status of what we found so far. So if you had to visualize everything, this is what it would look like in our end effort. A lot of driving, a lot of sampling, a lot of man hours. And to accomplish this, this is not a task for 3 to 4 people. This is a task for a large army. Luckily, we have a great crayfish working group over here in this part of the country. So we tapped various universities and people to help us. Sue John out of Longwood University and his crew. Paul Cabe and his crew out of Washington and Lee University. Zach and his crew out of West Liberty University. He has a small army in itself, and here's Bronwyn, Dave Lieb, and myself included in there as well. And then what ended up happening a lot is just everyone. So we would have individuals from all the different groups come out and assist. And what this eventually led to was 1,283 sites sampled across 6 states, including South Carolina, North Carolina, Virginia, West Virginia, Maryland, and PA, with a genetic data set that's comprised of nearly 1,750 individuals. However, we are here today to focus on Virginia. So in the state of Virginia alone, after the initial survey efforts, we have 280 sampled sites. Again, here's a visualization for it. If you would like to see it at a watershed level, here's a Huck 4. Here's a Huck 8. And then here's the Huck 10, which is what we were keying in on. So again, focusing on Virginia, here's everywhere we sampled within Virginia and its watersheds. And to kind of better visualize this, you might be saying, well, there's all your sites, but where'd you get species C? All these colored green dots are species C sites. I believe yellow, purple, and orange are also species C, where we had other species, other interesting species occurring. Blue's kind of our wish list sites, which we would like to go back and hit up some extra spots. Anything with a skull that is gray is a site where we got no crayfish despite sampling efforts. And then unfortunately, anything with a skull and a colored background is an invasive species occurrence. So we did all this. We're looking at the genetics. What does it all mean? So here is a circle tree. Some of the text on this is very small, so I've kind of expanded out here on the side. So you might remember earlier that hystricosis was originally thought to be very closely related to spicatus. Well, spicatus is down here. Hystricosis is way up here. They are not even closely related. So what we have going on here is a lot of going away and converging back to a certain body plan, depending on the environmental stressors. All the aforementioned previously listed species kind of clayed out nicely. Hobsorum's off here on its own. Have Aldermanorum here, Spicatus here, Acuminatus here, Johni, and then Franklini. But then what are all these other things? Well, these are all clades and branches that are at this moment potentially undescribed species from what we found. Anything with an asterisk occurs in Virginia. Anything with an asterisk and a plus mark occurs in Virginia as well as other states. So even if you looked at this extremely conservatively, we're looking at least 6 potentially undescribed species in Virginia. However, this is just based on genetics, so we are going to go back and confirm through everything else needed to describe a species. Biogeography, morphometrics, the whole 9 yards. Each of these, if formally worked up into a species, will have all of that. So here are some of the original body plans of the animals that are already described. And here are some of the new body plans of things we were finding. As you can see, a lot of wide variability, different color forms, muted colors, highly contrasting colors, narrow areolas, wide areolas, almost obliterated areolas. Some are plain patterned, others have this highly camouflage pattern. Some have saddles, some have lines. We have one that looks entirely like a acuminatus with bright stripes down its tail. It's just not pictured here. So there is a lot of variability, and the cool thing is these characters hold throughout the drainages that they occur in. So this isn't where you would find all 6 of these within one little drainage and it's just this wide cline. These forms are static and hold throughout the drainages they occur in. And it kind of becomes chaotic to think about all this because there's so much evolving away, converging back into the same body plan. But if you read Gunter's paper about color morphs and patterns and how all this happens, everything kind of comes together really nicely. And it makes a lot of sense when you look at it from all the different aspects. So we're kind of excited about this, but there's still a lot of work to do. Again, conservatively 6 potential species right now undescribed in Virginia alone from this complex. We're going to have to hit it with a more fine-tooth resolution to confirm. This involves additional sampling, additional genetic analysis with RadSec and PopGen, and then morphometric confirmation. In regards to conservation needs, some of these species will be very common throughout the state and will require little to no conservation needs. Others may be very narrow range endemics and may require immediate conservation efforts. And it's not so much human impacts we're worried about so much as invasive species. Currently, Virginia has more introduced and invasive species of crayfish than any other state that I've worked in. We have the big 3. We have Virilis, Rusticus, and Clarkii. All are well documented and noted throughout the state. However, Virginia has this fun guy of Faxonius ozarki. It was first noted by Loafman throughout the James in the early 2010s. And since then it has exploded. It's everywhere in the James. We recently found it in the Roanoke and it's really starting to take off there. It gets almost as big as Virilis, and they really just, once they're in an area, the population really takes off and they just fill in all the gaps, push the natives out. In addition to this, you also have Christovarius and Obscurus that occur up in the mountains. Some of these species occur natively in the state, but in the mountains they're not supposed to occur there, so they've been introduced through bait bucket. So Virginia's just kind of under this attack of invasives. And again, if you had to visualize it, remember all the skulls are invasive dots. And even through here, even though it looks like there aren't a lot of invasives, there are. It's just that not all of our sites contain them, but we have them from nearby sites as well. With that, I would like to take a minute to thank everyone. As you can see, this was not a small project. This involved numerous people contributing their time and effort from various agencies, private entities, and groups. I would like to especially thank the landowners that didn't shoot us. We got a bunch of really funny landowner interaction stories out of this. And with that, I conclude my talk and I will take any of your questions. Thank you.